My name is Alexander Opitz. I'm assistant professor in biomedical engineering at the University of Minnesota. And we are gonna host a computational modeling workshop in non-invasive brain stimulation today and tomorrow. And yeah, as many of those online conferences now all popping up, we had also initially planned to have an in-person meeting at this time. It's one time of the year where it's actually quite nice in Minnesota. We had. I want to just give a bit of a overview lecture. So what's kind of for you in store in this workshop, or why we why we're gonna want to do modeling in non-invasive brain stimulation. And so we're gonna focus on two main modalities. They're kind of displayed here on that slide. One is on the right, a transcranial magnetic stimulation, or TMS, on the left, a transcranial direct current stimulation, or tDCS. So those are both methods that are applying a stimulation device on the outside of the head. So that's why we call them non-invasive. We don't at least make a hole in the head to implant some electrodes, but we're trying to deliver electricity to the brain in a manner that keeps the skull intact, or the general integrity of the head. And so yeah, those are basically the two most popular methods used in the field. And this is some like say most of the focus in of this workshop. Obviously a lot more than those two, but those two, say, work with electricity. Those are ones we're kind of focusing on. So what is TMS? So I think for many of you, obviously you're very well familiar with that. But I think also several people have signed up for that. They might not be as familiar with all those methods, so kind of quickly shortly summarize what they do. So TMS, or transcranial magnetic stimulation, there we have a time-varying magnetic field that's being created through some coils, a magnetic coil that has some wire loops that you put somewhere over your head, the time-varying magnetic field, and that induces an electric field in the brain underneath the TMS coil. And TMS, with that you can create quite high electric fields, somewhere in the range of say 100 volts per meter, that are strong enough to initiate action potentials in underlying neurons. And so you have also some, for example, a direct measurable output, something like a motor evoked potential. So if you stimulate the motor cortex, you can make your finger twitch, and that's something you can measure. The other methods, transcranial direct current stimulation, or also other forms of that, for example, transcranial alternating current stimulation, where you either apply a DC electric field or AC electric field. How does that work? There you have, you can imagine that a little bit as some inverse of EEG. So you have some electrodes that you attach to the scalp, and through those you apply those weak electric fields. Some of those, everyone who's, yeah, please mute yourself, or also even I think you can also mute everyone else who's not muted themselves. Yeah, everyone, so going back, so for those methods, you apply those weak current through this electrode attached to the scalp and have some more like more subtle electric field in the in the brain, where those effects are more indirect. So those are those two those main methods. And so one issue we have a bit with those, it's a bit of a problem of a black box. We basically apply something on the scalp level, but we have some like a hard time to directly know what's going on in the brain. So, for example, for transcranial electric stimulation, we apply those currents on the scalp, and then the question is, where do they go in the brain, or what's happening? And there's something we don't typically have directly access to if you do human experiments, because we cannot, like, say, implant electrodes and measure how strong those fields are. There's still certain things you can do to try to measure those currents, and I think we're gonna hear about those in later talks. But from, say, most typical 99% of applications, we don't really have access to that. And I would say modeling can help shine some light in this problem. We have, say, two main issues we can help with addressing using computational modeling. One is the issue of, say, targeting. I would say talk more about that. Where do electric fields go? And the other issue, say, like of dosing. So how strong are those fields? And yeah, for example, also directional information. So why do we need targeting with those methods? So for TMS, you have a certain, say, spatial resolution that comes with that method. That is something that's pretty obvious to, if you do like a so-called motor mapping experiments. We have TMS codes over the motor cortex and move that around in centimeter steps, and then measure motor evoked potentials. So you can get something that called a motor map that's somewhat shown here on the right. And so what you see is, if you move your coil, say, like a centimeter or two, then you can have a very different physiological reaction to that. So you see that it will make a really big difference where we place a TMS coil. Obviously, if you have a direct nice readout like MEPs, that's something where you can use that to guide your stimulation. But for other brain regions, you will not have such a direct readout. For example, if you stimulate over the prefrontal cortex, that's like a popular target, especially for psychiatric application, will not have such a direct readout. And so then, how do we then place our, say, TMS coil in this type of situation? Yeah, this is here an example of all those colored dots here over the prefrontal region. This have all been target regions in depression studies of TMS, and you see there's really quite a bit of spread in those targets. And to me, it's not surprising that there are like some different outcomes of those studies, just given the big variability in those targets. And so people are hoping that we maybe have a more, say, precise, model guided application of those TMS applications that can potentially help improving the application of that method. Then, where I think a second very important issue where modeling can help is for dosing, is basically determining, say, the electric field strength, but not only the strength, but also, say, orientation, and all other related things related to dose. So why is that important? So from, say, recordings, so here I'm going to just quickly show those are some of, we did single unit recordings in non-human primate motor cortex, where we applied tDCS stimulation at varying intensities, 0.5 milliamp one and 1.5 milliamp. And what we saw, it's like a really nice dose response that with increasing intensity, single units got increasingly more entrained. With entrained, we mean like alignment to the peak of the stimulation. And basically showing like a real direct relationship between electric field strength and physiological response. And so, and this is something, you know, that's the very easiest thing to, you know, manipulate experimentally. If you do TMS, you just, you know, have your knob where you can adjust your stimulation intensity. If you do electrical stimulation, you just set, you know, your stimulation intensity of, say, 1 milliamp, 2 milliamp. People are already looking to do even increase that range, but then the question is like, which intensity should we choose, right? So here, if we choose between, say, 0.5 or 1.5, we would have a very different physiological response in that case. But for human application, that's to be not something, you know, we can directly so measure as we can do in those animal experiments. And this, I think, also another important part where modeling can help. And finally, something I would say the big issue, you know, we have in the field is individual variability. This is most often drawn into individual variability with respect to, say, outcomes of stimulation, both with physiology or behavior. But we also have just lone variability just based on, say, electric fields. If we apply 1 milliamp tDCS across many different people, will have quite a few differences in the electric field strengths. And so I myself, or like other groups, have done invasive measurements, and they have, where they say we have really, you know, can be more sure about what we're getting. They also have seen there's really quite a spread, at least they would say a factor of 2 differences in the electric fields that we're getting if you apply those methods. So that's also have the hope that we can control dosing and like tailor that to the individual to have a more, say, accurate stimulation. So where are those differences coming from? So one thing is, you know, individuals have different anatomies, head or brain anatomies, for example, clarification, CSF thickness, or skull composition. So those are all things that can influence electric fields in the brain that are being induced by those methods. And there we had many different modeling papers about that. For example, just mentioning some of those, one of our earliest paper looking at different factors that influence or shape electric fields for TMS. One thing I just want to highlight also is someone like makes a real difference between coil orientation. If you compare on the right side, from a parallel versus perpendicular, or you see like the difference in the color coding. That also the target, the main target, the focus of electric field strengths can differ quite dramatically between those coil orientations. Also for tDCS, we looked at different factors. So on the very right thing is some basic example of tDCS current flow. So you have some more current coming from those electrodes. A lot of like shunted in the skin, but those ones make it say radially through the skull, but then will align again parallel in cerebrospinal fluid, and you can get like pockets of hot, I will say, have those red colors of high current densities, and that also leads to stronger electric fields in the underlying brain regions. See on the left side, we have some highlights where we say like over like CSF is thin, it's not a strong CSF layer, we'll have a strong electric field. And so those are things we can try to account for, and that using modeling. And so one popular method for that is finite element modeling, and so that's where we have a lot of more to come in also talks after me, or those demos. So just not gonna go much into detail, but basically we have anatomical MR images that we create a FEM model from it. So we're starting with the T1 image, then segment tissues, and then do create those FEM models from that, and then run some those numerical simulations to say estimate the electric field distribution. And so this is something we've been starting to develop like 10 years ago. But that was really quite an software engineering challenge. But this has really matured a lot lately, and they have our open source software package, NIBS, where we hear a lot more and have walkthrough and demos later today on both days. And so there's something we get recently NIH funding for that, further develop that, also host those workshops to kind of popularize the software more. And so there's more to come. And so right after, later today, we have Axel and his team will talk about it, basically how we go from this MR image to those simulation results. So stay tuned for that. They talk about also electric field components and all those technical details. On day 2, we have planned a bit more, say some advanced modeling. So one thing also my lab is very interested, but also others is somewhere studying effects of non-invasive brain stimulation in animal models, because they can basically have more like a larger tool set for neuroscience measurements that you have can apply that you don't have in humans. But at the end of the day, we want to have those obviously apply in human applications. And so I think what one has to be really careful is like how we translate those protocols from animals to humans, because if you do say 1 milliamp tDCS in a human compared to say a monkey or mouse, you will have very different electric fields. So this is kind of shown here. First, some of the spread is a bit different, but also field strengths, and this will also always differ from montage to montage, and so that's something I think people have to be very very careful about, because sometimes you see very strong claims that comes there. We did some measurements maybe in a mouse model did not necessarily translate, like say one-to-one to humans. And I think for that is also modeling can be very useful tool set. And so also for that second day, we are also gonna share also all our current animal FEM models that we have, so we can have a link for you to download, so you can then also just start using that for your own research if you're interested in. And then we're also gonna give some pointers to say applications There are also say maybe non-standard in SimNIBS, for example, a case of here, this is like in pediatric stroke. People are like really interested in using non-invasive brain stimulation for stroke rehabilitation. But if you have, say, for example, really, extended lesions in your brain, that will have quite an influence on those electric fields. So also trying to model that. So this is something which is not like automated, but we'll give some pointers how you can do that if you, for example, add like manual segmentations of lesion volumes to your model. So this was somewhat like a bit the overview of what's to come in that workshop, and that's just very shortly to see. I think that what is called the future of modeling in that field, and so try and also give a bit of some talks in that direction. I think one say very hard or important topic is somewhat like doing multi-scale models, say bridging, say the physics with the physiology. Obviously, if you have, an electric field is somewhat like a zero order model of your physiological response. Obviously, it depends a lot what neurons you're targeting. And so on. So that's something people starting to address using multi-scale models. Also, one thing we've been really interested in is modeling plasticity. A lot of our applications, clinical applications, some like say repeated application of the stimulation, the hope to make a lasting change in brain activity is something. Don't really have many models for that, or say physiologically realistic models. That's something we're going to do. And then the other part is some integration with imaging. So for multi-scale models, we have also here a bit more. We have a neuron that's being exposed to those electric fields. We'll see it's also a bit to some bridge that between quite different spatial scales, so like this, the large scale FEM model, and then like a very small neuron in there. So that's something my lab is working on. You see, just see on the left side, just a very big difference in coil, or say coil orientation or electric field orientation. So for example, depending where they have some current coming here from left to right, we'll have, say, depolarization at one side, hyperpolarization on the other. And if you somehow rotate it by 90 degrees, you will have a completely different polarization of that neuron. And this is something we can expect, or we know that will make a very big difference in physiological response. And this is something we can often control with brain stimulation for TMS easily with, say, coil orientation, if you have a figure of 8 core, but also if you do, say, tDCS, depending where you place your electrodes, it's always at least 2 electrodes. You can also shape electric field direction and optimize that. So that's something I think those models can be really useful. And then also moving to then modeling of, say, RTMS, if you have, say, repeated pulses, and so there's a where you have somewhat like a history of activity, and not only just some like a snapshot of activity. And so there I run some simulation. We see depending on the intensity, don't always get an action potential initiated for every pulse, because there's like a history of the previous pulse, and that can also make an influence on the effectiveness of those protocols. So towards the end of the second day, we'll have a bit some more outlines for those type of effects. And somewhat as a final, we see also the future of those methods, some like integration with imaging. For example, integration with functional magnetic resonance imaging. If you like, for example, one thing that I did is people now really starting to look also not say anatomical targeting, but also network-based targeting, and you can estimate your network using fMRI, for example, using then TMS electric field as a seed region for functional connectivity, and determine the functional connectivity profile from that electric field seed region shown here. And that's something you can, for example, use for DLPFC targeting. And this one modeling study I did a few years ago, where we looked at different potential targets in DLPFC regions, and showing that we have different connectivity profiles depending on the target. And it's also something I think, depending where you place it, is maybe more likely to have one different target engaged than another one. And it's also something you can potentially use for optimization, or how to guide your TMS application. So yeah, that's was kind of my overview over that workshop. So I think we have a lot of exciting talks or demos right before us, and just want to just give a few quick acknowledgements right now when giving the talk. So first, want to really thank my lab for helping organizing that workshop. For Ivan, has been he's the Zoom master, so he's taking care of all the Zoom stuff running. I hope that runs smoothly, and organizing. Also, Katie has been involved a lot with speakers and a lot of organization things, but also Sina will also be giving a demo and a talk, but also everyone else been really working a lot to make that workshop happen. Also, thank all the speakers who have agreed to do their presentation online, to be really flexible for making the switch from coming here, some flights already booked that or canceled, and now do this all online. Or thank you for that, and also thanks for some of the funding agency that also they partially sponsor the workshop. If you're interested more about our own research, you can also look on our web homepage. We have some more examples. You can follow us on Twitter if you want to tweet about the workshop. Is also something we will welcome. Okay, I think then I stop here. Then I think if you have some questions at this point, I'm happy to answer a question or two.
Thank you, Alex, for the great talk and for beginning of the workshop. Now, Alex, the question is, you showed the results of spike entrainment by tDCS. Did you check if phase lock-in correlates with local field strength?
Right. I guess you could also answer that. I guess we looked at that, but I think we did not see a clear relationship between, say, between the local field strengths at this particular neuron with the level of, say, spike entrainment. And I think the reason is, I mean, so like in general, so there's also preprint about that. I mean, also paper now, like in a third round in revision. So hopefully it also comes out at some point soon. But we had like about, say, 10% of the recorded neurons that reacted quite strongly in this type of, say, entrainment. But we also had a lot of other neurons that did not respond in this, say, entrainment type of way, but I say had some type of a different response. But yeah, to keep short, we did not see, at least in the individual neuron level, did not see a clear relationship between, say, the local local field and this entrainment. And one thing I would really like to know is somewhere like, what is the orientation of those neurons from which we recorded from? That's something we just unfortunately also don't quite know which neurons that are. I mean, they just basically put those electrodes there and you record from everything you can get. And yeah, I think they're just different type of neurons that have different orientations or different type of, say, you know, physiological properties that somewhat determine the response. Also, electric fields don't differ that much over the somewhat localized area, so it's not, yeah, not that variation, not so much variation there.
Thank you. Also, I remind to everyone that they can write questions in chat at any moment. And until we are waiting more questions, I can have one from myself. So you called your talk "Why to Model." Thanks. So Alex, do you think if there is a reason why not to model, or in what cases we shouldn't rely on model that much?
No, I don't know. I mean, it's, you know, it's just one thing. I guess you just have to be also careful what you get with the models and how you interpret that, and you know, like know the limitations. I think that's also, I think model validation and accuracy is very important. I think we have just some talks coming up about that right now, and so that's why also, you know, starting basically doing only modeling and someone moved a bit and doing those experiments, also because I wanted to know basically what the model, say, predicts as electric field. Is that really something you can actually measure and you know, get with a real life experiment? And so that's why, yeah, measurements are very important. And you know, if you can measure something, measure it. But I think that just it goes hand in hand. So if you model can make prediction, you can measure that, or vice versa. From your models, also somewhat like of a framework we can, you know, have like develop a mechanistic theory how those brain simulation methods work. I think for that it's useful, but not like, you know, if Simnips gives you an output, it's not something overthink that there's like somewhat like a large error bar coming with all those model results. So if you say it's 0.5 volts per meter, maybe it's could be 0.7, could be 0.3, I know it could be 0.2, but it's probably not like, I would say the order of magnitude certainly correct, but you know, it's not like whatever, you know, 10% precision. So that's more bit of caution.